I'll give you a short introduction. We are pleased to have Wei Dai for our tomography section for the winter EM course. She's beginning this section with an introduction to tomography. Wei Dai comes from Rutgers University, where she not only uses Cryo-EM, but Cryo-ET and CLEM to look at the structural biology of neurodegenerative diseases and phage virus maturation. So I'll let you take it away.
Okay, thanks. So I'm trying to figure out how many students are in this session with me. So I'm happy to give the opening lecture on tomography. So the title will be Introduction to Tomography. So what the purpose of this lecture really is to deliver, to introduce to you the fundamentals and also language in cryo-electron tomography. So in the future, you can make an educated decision if this bioimaging tool is useful for your research. But before I get started, I'm wondering, is it okay for everybody, for students who sign up for this course, can give a 20-second introduction of yourself, self-introduction, basically your name, your position, and your understanding or your experience with cryo-electron tomography on a scale from 0 to 5. 0 meaning never heard of it, never used it. 1 means you read at least a couple tomography papers. 5 means this is one of the major tools you are using in your research.
Okay, I'll step in as moderator and we'll go semi-alphabetically based on the participants. So I'll call on Andrew.
Hi, I'm a postdoc and I use primarily single particles. So I'd say 1 for tomography.
Thanks.
And Amal?
Hi, this is Amol. I'm also a postdoc at Rockefeller University. And I would also say my experience is somewhere between 0 to 1. I also use like mostly single particle.
I see.
Okay. Wang?
Hi, it's Wang here. So I'm a postdoc at Boston Children's Hospital. So I heard of tomography, but I have never done that. So I would say I'm never 0 for that.
Okay, Agata?
Hi, I'm staff at MSK, and my experience with tomography is 0.5. I've read some papers.
Okay. Okay, that's everybody, right? I also got a person in chat who has mic problem, but basically is fundamental, very 0 to 1 in tomography. So thank all of you for this information. So for this lecture, I will more focus on introduction and lecture instead of, on discussions. Okay, let's get started. So I always, since the pandemic, I always start my lecture from this New York Times article featuring several major scientific discoveries on this particular virus from cryo-electron tomography and cryo-EM. So let's see. So the first one I'm showing you here is a 3D map. Can everybody see my cursor? Anybody?
Yes.
Okay, thanks. You can see a 3D reconstruction of the particle. You can see the spike protein decorating the envelope. And then at the background, it's a slice of a tomogram. And you can see basically from the cut away view to see the genome packed inside the envelope. Okay, so this particular research is done by tomography. And then the second is probably most people write this paper already. It's a structure of RNA-dependent RNA polymerase. But basically, this research is done by cryo-EM single particle analysis at very high resolution so that you can understand the molecular mechanism of how the drug interfere with function of this enzyme. For understanding of that molecular mechanism, you need high resolution, and this is from single particle analysis. Number 3 is 3D structure of the spike protein on the membrane. this is also very high resolution structure reconstruction, and understanding of this structure help us to design the vaccine, to design antibody therapy, and also to understand the molecular mechanisms between the spike protein interaction with membrane receptors. Again, this is from single particle analysis. So the last one, you can see this is a movie of a subregion of a cell that has been infected by various particles. And then it shows how the various particle can hijack the endomembrane system as dedicated site for viral assembly. So from this, you probably can know, this is also from tomography study. So out of the 4 samples, I showed you 2 from cryo-electron tomography, 2 from single particle analysis. Both of the techniques use the same type of cryo-electron microscope. But in terms of sample preparation, data collection, data processing, they probably use very different routines. So if we look at map deposited in EMDB, it's very obvious. Single particle is more mature than tomography, because if you look at the map, tomography plus subtomogram average, the total map is only about 13%, versus single particle is over 80. Okay, so single particle is more mature, and there are reasons behind it. I hope by end of the lecture, we can have a discussion. Why? What is the limiting factor in cryo-electron tomography? Okay, so today I'm gonna, first of all, use a couple slides to summarize or to introduce what is cryo-electron tomography. And then I understand all of you have learned a lot in sample preparation in single particle modules. So I also want to cover a little bit about sample preparation, but only special aspect relevant to cryo-electron tomography. I'm also going to very quickly go through the workflow in terms of data collection, alignment, and reconstruction, focusing on tomography specific aspects. So lastly, I'm going to give you one example to go over the overall workflow to see how can you combine cryo-electron tomography with other bioimaging modalities, including FIP, correlative light electron microscopy, to understand cell biology. Okay, so that's the outline for today. What is cryo-ET? So you all learned single particle analysis already. So basically the overall workflow for single particle, several things. First, you probably want to purify your protein complex, and then you want to image thousands or millions of particles. Eventually, you computationally combine structural information from the thousands or millions of particles, eventually to give you a 3D map. So that 3D map usually has very high resolution if your sample is stable, but usually we see it as an average of structures of thousands or millions of particles, right? So it basically represents the common feature of your sample. On the other hand, cryo-electron tomography, sample for this technique, it could be purified protein complexes. It also could be, for example, an acylated organelle, enriched membrane fractions. It could be a bacteria. So it's basically, it doesn't have to be homogeneous. And then the technique to understand it is basically like CT in a hospital. You load the sample into microscope, you take images from different tilting angles, and eventually you go back to a computer to computationally align those images and back project them into a 3D map. Okay, so this is the fundamental of cryo-electron tomography or cryo-ET. So one thing to think about is look at a 3D map. It's a 3D reconstruction, but using information collected from this protein complex itself. So it's not an average of thousands of particles. It represents a very unique object. So because of that, we can say for cryo-electron tomography, we can handle sample with a unique structure we can say we can treat particles one by one. And also we can potentially study the structure of the protein complex in a complex cellular environment, like the earlier example I showed for the coronavirus assembly inside the cell. So far, any questions about tomography? Okay. So sample preparation, as I said, you learned a lot in your single particle modules. So basically, sample preparation meaning you want to keep your sample in vitreous eyes so that it can be imaged in the very high volume in a microscope, right? So it's the same philosophy as single particle and sample preparation. You want the sample to have good concentration. You want to make sure the grid has good ice thickness so that you can have good contrast. But one thing probably special about sample preparation in tomography is, very often the sample is not purified. You will eventually need to track your target either in their native environment or in a very complex environment. Because of that, for this particular factor, this particular aspect, you need to think about it at the sample preparation stage, okay? So plunge freezing is still the most common sample preparation method for tomography. So for samples for tomography study from, for example, this is probably purified complex, some phage particle or variparticles. This is lipid vesicles or maybe liposomes or purified endomembrane system, organelles, or microsomes. And this B is very obvious, is a bacteria. And A is a eukaryotic cell, probably. But for all these type of samples, potentially you can use plunge freezing to prepare for them, okay? So, but from B to F, very often you process them the same way as you process a single particle sample, basically apply the sample to EM grid and plunge freeze them. So for mammalian cells, it's slightly more complicated because they're bigger. And also, especially for some adherent cells, you have to let them adhere to a matrix for them to keep their physiologically relevant morphology for structural studies. Because of that, we often culture those cells, or we call them plate the cells on EM grid before we plunge freeze them. So because of that, you have several factors you have to consider. For example, what type of grid you want to use, and then what kind of mesh you want to pick. And then usually also have to make the decision of supporting films. We want good support to give, for example, continuous carbon, or you care more about contrast, so you can use quantifoil or holy grids. And then also you have to consider how well the cell will be able to adhere to the bottom. Do you want to add in, for example, coding proteins to help them adhere to the bottom even better? So in my lab, this is how we set up. So we use those special 35 millimeter glass bottomed Petri dish. We put, I hope you can see it. We put one EM grid of our choice at the bottom, and then we plate the cell into this little dish. And then we let them grow for a certain time. And then we plunge freeze the grid, and then we can take the grid for imaging. So here in this panel, I'm showing you how the cells look like. So for this particular one, you can see the cell body and you can see the lung processes. They adhere to the bottom quantifoil grids pretty well. So the region and then the lung process, actually, you can directly image them. Okay, so any questions so far? Okay, so now let's say you have your favorite sample prepared on EM grid. Now you can load the grid into the microscope for data collection. The raw data you collect out of a microscope is called the tilt series. Okay, so this particular is a tilt series of membrane fraction from a yeast. So if you use the edge of the hole as a reference, you can see basically this is a set of image taking of the membrane fraction from different tilting angles. And then the tilting axis is probably over here, right, along this line. And then the images are a little bit jumping around, not too bad. So the jumping around is caused by the stage shift during tilting, okay? Then, after you put the sample into the microscope for data collection, you actually have a lot of options, meaning you have to make decisions. What is the best for your particular project? Some of them are gonna overlap with your single particle project. For example, defocus. So you have to pick up a defocus to balance contrast versus resolution. I hope this is very obvious for all of you after the single particle modules. And then also total dose, right? How much electron you want to give to your samples. So this is a balance between signal versus radiation damage. You want to see clearly, then you should give higher dose, but you are running the risk of radiation damage, right? And then one special aspect in tomography versus single particle analysis, let's say after you determine the total dose, you can apply it to the sample. You also have to determine the tilt range and tilt increment. Tilt range basically means what's the tilting angle, tilting range, highest and lowest. But this is probably determined, most likely determined by the mechanical restriction of stage. For most microscopes, you can go from -70 to +70. But there are many things we can discuss about, for example, tilt increment. For example, you know the total dose your sample can tolerate. You need to divide the total dose into images, the number of images in your tilt series, right? So theoretically speaking, the lower number of images in your tilt series, the higher dose each image can have, the higher contrast and signal you can have in each image, and probably gonna give you better alignment and better map. But at the same time, if the total number of images in your tilt series is too low, you are going to miss a lot of information between the two neighboring images in Fourier space and in real space, right? So this is a factor. This is a decision you have to make. Basically, mostly ultimately determined by goal of your project. It's a project you just want to see clearly of your sample, or it's a project you're actually hoping for a subtomogram average map. Because of the different goal, you're probably going to make different decisions. And also your sample geometry is a very important factor to consider. How big the object is, how small it is, what shape is it? Is lipid vesicle or is a membrane pieces mostly laying down on EM grid? All those factors you need to consider before you can make a decision what imaging configuration you want to use for your project. So I put this as concept check question. We can talk about it later, but if you have any question relevant to your project, I can always stop here and then we can talk about it. Okay. You can just unmute and speak during the lecture if you have any question. Okay. Okay, so once you collect your data, you need to transfer the data to a computer and then process it from 2D tilt series into a 3D map, right? So to do that, you have to align those images in the tilt series to fix, for example, XY shift. Basically, that is the implant X and Y translations. And then from this raw data, you kind of can see where the tilting axis is, right? But the rotation of the images in the tilt series usually is not perfect, also because of the mechanic imperfection of the stage and then the goniometer. So that is something parameter you might also want to consider to polish. And then each image in the tilt series represent some structural information of your specimen from a particular orientation. That orientation is defined in the tilting angle of the image. That parameter, you also want to incorporate it into your final reconstruction to get a reasonable map, 3D map. And then just as single particle projects, you also want to correct the focus of individual images so that for you to be able to see your structural features more clearly, okay? So this, I'm showing you the map, the raw data before correction. I'm sorry, this is and this is after. So they're not jumping around anymore. And you can see the tilting axis is better defined after we fix the slightly imperfect rotation, right? And then from this data set, you will be able to back project computationally to generate the 3D map. Okay, so this I'm showing you slice view of a tomogram reconstructed from the tilt series I showed you in my earlier slides. So you can see basically that's membrane pieces, plasma membrane. And then from those membranes, you can see individual like little donuts or little flowers. So those are my research interest. Those are glucan synthase, fungal glucan synthase. They are the membrane protein to synthesize, catalyze the biosynthesis of glucan. Glucan is one of the major scaffold component of fungal cell wall, okay? And also this particular enzyme is also the drug target for echinokindin drugs. Okay, that's why I'm interested in it. You can see they're still in membrane embedded form, and then they seem to form clusters, right? The little donuts, little flowers. Okay. And then from there, you can do many things. Because glucan synthase is still in this particular project, we didn't purify it. So it's still in the membrane embedded form. So you can analyze their native distribution and then compare different cells with different genetic background, for example, or different culture conditions, for example, drug treated versus control. And then you can actually quantify their distribution and then by plotting individual coordinates, right? And then also you can perform subtomogram averaging by computationally extract each flower. We call it a subtomogram. And then computationally combine information, align them and combine information from hundreds or thousands of particles to eventually give you a 3D subtomogram average, as I showed in the movie, the purple map. Okay, and then this resolution is probably about 14 or 15 angstrom resolution. At this resolution, you will be able to annotate different components and to see how different domain of the protein complex interact with each other. Okay. But, let's think about subtomogram averages versus 3D maps from single particle. So for example, this the top plot is a resolution plot of 3D maps deposited into EMDB, resolved by single particle analysis. You know, it seems like if the sample is stable enough, the peak is about maybe 4 astron resolution. But this, the bottom panel shows you the distribution, resolution distribution of subtomogram averages currently in EM data bank. So I would say the resolution compared to single particle is a lot lower. It's probably from 10 all the way to even 40. So this is something for you to think. Why subtomogram averaging maps, the resolution is much lower compared to your 3D maps from single particle analysis? Anybody want to comment on this? Okay, we can also talk about this at the end of the lecture. Okay, so we talk about sample preparation, data collection. Next, we are going to move on to application of cryo-electron tomography in cell biology. Because cryo-electron tomography allow you to resolve the structure of the protein complex almost in their very native environment. And then literally you can say you can capture the structure of the protein complex when they are performing their biological functions, for example, inside the cell. So this type of research, nowadays, people often call it structure cell biology. So for example, I'm here showing you a cell plated on EM grid, as I introduced earlier. And then I use the lines, the dashed lines, to show you approximately where the nucleus is and then where the cell membrane, plasma membrane is. So for example, at the cell periphery regions, you can almost clearly see the hole underneath, right? The hole on EM grid underneath. That means those regions are transparent to electron beam. Basically, they're imageable directly by electrons. But most of the region, the cell body, and also especially the nucleus region, you actually can't see much. They're very dark or black. So most of the region, they're too thick for electrons to penetrate. So we need to solve that problem for us to have a successful cellular tomography project, right? So the tool people currently use is called focused ion beam. Okay, focused ion beam application can help us to solve the thickness problem. And then second, the cellular, cytoplasm is a very complex environment. You have proteins everywhere, complexes everywhere. And then how can you carefully or very confidently say that particular density is the complex of my interest? For you to be able to track down your target in very complex cellular environment, the new technology called correlative light and electron microscopy, okay? So the two challenges for cellular tomography is one, thickness, number two, target tracking. To fix the thickness problem, we have a technology called focused ion beam. To fix the second problem, overcome the second challenge, we have a technology called correlative light and electron microscopy. Okay. So next, what I'm gonna do is I'm gonna use 15 minutes to use one example, a recent publication from Elizabeth Wheeler's group, to show you the workflow of cellular tomography project, and then how tomography can be combined with FIP and CLAM for you to be able to resolve structures inside the cell, okay? So for this project, their target is called LARK, or leucine-rich repeat kinase. So this protein is a very important target in Parkinson's disease because it's the most mutated gene in familial PD. And then this protein definitely has a lot of important functions in neurite growth and neurodevelopment. And then it has binding affinity to microtubules. And then some certain mutations and treatment with drugs can to inhibit the kinase activity can actually increase the binding affinity of those LARK to microtubule, basically recruit them to microtubule association. And this protein is very important, but the full-length protein structure is not available because of, maybe challenges in purification or the complex domain organizations. It's a multi-domain protein made of multiple domains and then connected with certain linkers. Some of the linkers are very flexible. Okay. So the approach they took is to put a fluorescence linker or tag to the full-length protein and then express the protein in the cell and then play the cell, use CLAM to identify this protein on microtubule, and then find where the microtubule is, and then to perform cryo FIP to sync the cells so that you can image the microtubule, intracellular microtubule. Eventually, from those tomograms, you can perform all different kinds of structural analysis. So step one. So this is actually, for most of the cellular tomography project, this is probably the most important step, I would say, because you want to make sure your experiment is very well designed so that you can easily track down your target. And then very often people use, for example, overexpression to enrich your target so that you don't have a hard time finding them inside the cells. And then in this particular project, they also take advantage of a mutation has higher association with microtubule to increase the chance of finding those microtubules, okay? And then they overexpress those protein inside the cell, plate the cells, and then loaded the grid into a unit called correlated light and electron microscopes, CLAM. And then the picture I showed here is a Leica model we have at Rutgers. So your place, you probably have a different model, but the fundamental idea is very similar. It's basically a fluorescence microscope with a very cold stage to keep your frozen grid in liquid nitrogen temperature so that the grid is maintained in vitreous state during your imaging or screening session. So this is A, is how the grid look like. And B, they have a zoom in view to see the filamental structure, very likely microtubules that are decorated with fluorescence labeled LARK protein. Okay, so they get the cells ready and they perform CLAM for them to be able to track down where LARK proteins are. And then from there, as I mentioned earlier, that we still have a technical challenge because on average, the cell, when they're plated on EM grid, even for neurons or fibroblasts, they still pretty thick. So maybe 1 to 5 micron or even thicker, cell type specific. And then we know usually if you want to be able to see your sample clearly, you want to limit your sample to be maybe under 300 microliter, 0.3 micron. So to do that, we're gonna do FIP. I know you're gonna have a special FIP lecture given by Bill. So I'm only gonna show you how the instrument look like. for you to know, that's basically where you want to put your sample in. Your camera is at the bottom, but we also call it, very often we call it the tubing system because you have two bins. One is the SEM column for you to be able to visualize your sample. And then we also have the iron column for you to perform thinning of your cells, okay? So basically, this is the iron view of your cell, for example, before milling. It's pretty thick, right? It's like a thread egg, too thick for TM imaging. And E is after. You basically remove most of the volume above your target, and then below, also below your target, only keep a very thin layer of cell volume left. We call it lamella. And then F is basically from your SEM column view for you to be able to see what's left from that particular cell, okay? So again, this is before milling, and then you probably can tell this is still a very big, very thick area in SEM. This is a SEM image overlay with fluorescence image. And then this is the filamental structure, is the one they're interested in because they are large decorated microtubules. So C, I shows you after milling. Again, is the SEM image overlay with the fluorescence image to see where your target is, okay? And then this is basically the lamella generated from FIP. Okay. And then after you generate that lamella, what you do is you remove the grid from FIP and you transfer it to TEM. And then here, they look very similar, but this is actually a TEM image of your lamella overlay with your fluorescence image. And this lamella, you can tell, is slightly transparent, meaning the thickness can be penetrated by electron beams, right? And then use the fluorescent signal as guidance. You can collect tilt series of microtubules from this lamella, right? And then E is slice of a tomogram after reconstruction overlay with the fluorescence signal. You can clearly see really the microtubule features are really overlaid pretty well with the fluorescence signals. And then from here, you can see, you can do annotation, basically computationally annotate microtubules and then perform structure analysis, okay? And then again, see, I showed this slide basically to show you what type of information you can achieve from a representative tomogram. For example, I think this project is interesting because at this resolution, you can actually resolve polarity of microtubule. Okay, you can see which end is the positive end, which end potentially could be the negative end. And then you can also resolve protofilament numbers, okay? And you can perform all different kinds of structure analysis between large decorated microtubule versus control, basically no large associated microtubule. You can track individual filament. You can also compare, for example, distance between different filaments, and also what is the angle between neighboring filaments. So all this kind of structure and spatial distribution analysis information can be resolved from cellular tomograms, okay? And also, you can perform subtomogram average because, large associate with, a microtubule forming a helical interaction. So you can perform helical subtomogram averaging just as you can do with helical reconstruction in single particle analysis to understand how large interact with, for example, microtubules. And at the same time, you can treat this as like individual particles. Large forms dimers on microtubules. You can treat each dimer as an individual subunit to perform subtomal analysis and then resolve individual domains at this resolution. For the large domains with strong structure features, you can actually just directly resolve it and allow you to do fitting of those higher resolution or atomic resolution maps directly into the subtomogram average. Okay. And then in addition to subtomogram averaging, I also want to mention you can perform integrative modeling. Generally speaking, subtomogram average resolutions are still quite limited comparing to single particle projects. So what you can do at this limited resolution is you can use it as an envelope and then incorporate information about geometry, domain organization, and also taking advantage of atomic resolution structures from single particle analysis, from X-ray crystallography, and also from recently structure prediction, for example, AlphaFold, and then use those information, combine those into your subtomogram averages for you to be able to deduce more information, atomic resolution information of your structural complex. Okay. And then I think, the beauty of cellular tomography project is really, provide your channel to connect structures with functions. Using, one example in this is in the dimer, they were able to resolve one of the domain called WD40 domain. It's responsible for dimerization. So what they did is they performed the fitting and integrative modeling to propose a couple of key amino acids, for example, important for this interaction. And then they can perform genetics or mutagenesis. And then in the mutant, they can compare it with the wild type to see if mutation of that particular amino acids really leads to loss of dimerization and association with microtubules. And then they perform structural analysis, mutagenesis, and then later on, a fluorescence imaging to track down dynamics of LARC2 protein inside the cells. Okay, I think this is actually a very important application of cellular tomography for us to understand functions of your target. Okay. And then that basically that ends my first part of the lecture. So basically I spent 40 minutes to give you a very brief introduction of tomography and how it compared to single particle analysis in sample preparation, data collection, and then and also application. So I use one example to show you how cellular tomography can be combined with FIB milling and CLAM for us to be able to understand the structure and function of our target in native cellular environment. Okay, any questions about any part of my lecture? Everybody is very quiet.
I would want to really encourage the students, like if you want to show your video or speak, because otherwise you're not going to get the value add. We can gear the conversation more towards what's interesting.
Yeah.
Hi, this is Agata. So I have a question regarding what should be the buffer composition of your sample, because that would result in a different ice thickness, I would imagine. So, and then a second question would be, since you're collecting only like a
Okay. Thanks. Okay.
Images, then okay, maybe I should. Well,
Okay, repeat your question. Sorry.
so the first question is, what's the buffer composition that should result in the optimal ice thickness? And the second question is, since one collects only tilts of images, how many images should be collected to cover the entire Fourier space, or is the how much does the model suffer from the preferred orientation from the particles, since you only collect on a certain angle or certain amount of images?
Okay. So I actually really like your questions because I actually prepared. Also, I give you a reference that I used in my presentation. Thank you so much for all your questions. You basically asked like 3 out of the 6 list I want to discuss
All right,
in the
thank
second
you.
half of the lecture. Very
Awesome.
good. You can be my teaching assistant. So,
Thank you. I'm humbled. I'm honored.
so, okay, the first one is basically a sample preparation question about buffer. This is a very challenging one. And then I can only say it's sample dependent. If you're talking about, for example, purified protein complex, you probably want to use the buffer that the complex is most stable, right? And then if you're talking about, for example, purified microsomes or vesicles, you probably want to maintain a buffer, maintain it in a buffer so that they don't burst. And then bacteria, you don't want to put it in water, right? You want to keep them happy. And then in relationship to ice thickness, this is actually a very good question. Buffer can play a role in ice thickness. But again, as I showed earlier, very often this is a decision between several trade-offs, right? You want to make sure your sample to be stable. That's probably number 1. And number 2, you don't want the buffer to be too thick, sticky, that give you ice that is too thick, that you can't image properly anymore. One factor you need to consider in terms of ice thickness is for single particle analysis, you basically collect your image at 0 degree. The ice thickness basically is, this is the ice thickness electron need to penetrate and to generate the contrast, right? But remember in tomography, you have to tilt your sample. When it's tilted, think about the path the electron can go through. The ice thickness is actually higher compared to single particle projects, right? So ideally, you actually want your ice to be as thin as possible, will keep your sample completely visualized and embedded, right? But really about buffer, I can't give you an answer.
Okay. Thank you.
So what sample do you work on?
Oh, none, honestly. I was just curious how it's estimated and how people go about it. Yeah.
Okay, but that's a good one. So the image tilt increment is your second question,
Yes.
right? How can we determine the increment? So I actually have a slides prepared. This, right? We talk about the increment. I show you a figure over here. There are several things we need to consider. You also mentioned the missing batch. Did you? No?
Yes, I did.
Okay. Missing batch for single tilt tomography, there's not much you can do. Your microscope can tilt -70 to +70. That's it. There's nothing you can do about it. Okay. But within that range, basically about the increment, as I mentioned, first of all, you know the total dose that your sample can tolerate. And then the number, the increment step is basically determines the number of images in your tilt series, right? Bigger increment, less images, higher dose per image. But does it mean the lower number of images in the tilt series, the better? If you look at this, basically lower number of tilt images in the tilt series, meaning bigger gaps between two images, also meaning a bigger gap to compensate for in Fourier space.
Correct. So how do you then, so then you look for another particle that has a different orientation and may, well, like fulfill the gap?
I think, I can think of two things we can do. Number one is we do double tilt. So this is something people have been doing in the past, but it seems to be less popular nowadays. So for single tilt, you basically have a missing match. You have a batch of information that you're missing from your tilt series, right? So many of the microscope, I think most of the microscopes at NCCAT, you can actually flip the axis in the microscope, allow you to take another tilt series along Y, for example, because of, and then if you combine the two tilt series into one, the missing information is smaller. The missing range is smaller from a wedge probably to a cone, right?
Yeah.
Right. But can you think of any disadvantages of
The ice thickness might be different at the different angle, right?
anything else? Okay, let's talk about ice thickness in the two tilt. Number one, this is my phone. You're tilting this way. The thickness at a certain, for example, tilting angle, electron penetrate pass will be that, right? That thickness. And then double tilt, I mean tilting this way. This is actually a bad example because this, my cell phone is not a square. So it's not a good way to tell. But if I can draw over here, ice thickness is actually at a fixed tilting angle. Think about it, draw it on piece of paper. The thickness is actually the same in X tilt and Y tilt. And then, but the problem is, as I mentioned, you have a total dose tolerance, your sample, right? In single tilt, let's say you're taking 40 images, you divide the total dose by 40. So each image, you'll have, 2.5% of total dose. But when you do double tilt, you have to save half of the dose for the second tilt. That means each image only have half of the dose you can get from single tilt series.
So less contrast or?
Yeah, so each image look more blurry compared to, single tilt, but you do have a smaller missing information, smaller gap in that.
Okay.
And then this is from imaging aspect. There are also other ways for you to be able to correct missing wedge. I think, see, this is basically single tilt. You tilt that way. This is the reconstruction. You can tell basically you're missing a big chunk of information in that direction. And then this is the double tilt, the second tilt. And you combine the two maps, you can see the ball, the vesicle appear to be a lot more spherical compared to each individual tilt, right?
Correct.
And then the second way you can do is subtomograph averaging. Again, you raise a very good point. The example I showed from my lecture is membrane protein when they are embedded on membrane. So they do have the preferred orientation problem because you mostly see them top views. But let's say we're not studying membrane protein. We're studying, for example, ribosome inside the cell. And then here I use one of my friends, Muyen's slides, and then he likes cat. So basically you see each cat as a ribosome inside the cell. they are randomly distributed, or the orientation is random, pretty much random inside the cell, right? So each particle or each subtomogram has missing match from different orientations. If you combine them, eventually get an average, each subtomogram can compensate for others. Eventually you fix the missing match problem. Is
Thank
that
you.
clear?
Yes, very much so. Thank you.
Okay. And then, yeah, you also mentioned the preferred orientation. That's a major problem. That's one of the reasons that my subtomogram averaging of my glucan synthase cannot go further. Right. That's a very good observation. Okay, so anybody else?
I had a question,
Sure.
which you, I guess, raised during the lecture, which was about the resolution difference between single particle versus tomogram. So does it have to do predominantly with the number of particles that you have for averaging? For example, in single particle, of course, that's like crazy high versus what you have in tomogram averaging.
Yes. See, you are my second teaching assistant. That's exactly what I want to talk about next. Okay. You know what? Don't go. You start talking already. I will let you keep talking. What else can you think of? Anything? Particle
Also, like
number is number one.
for example, just the homogeneity of the sample, right? Like in single particle, you literally have just the particle alone and nothing else. And in the cellular environment, like you mentioned, it's like a lot of complexes, lots of membrane bound structures. It's just diverse kind of structures, and the patterns that you would get from electrons hitting these different structure would be different based on what it is, like membrane versus protein, or like
I actually really like this answer, but I don't know exactly how many, but I really like the one you bring up. So think about this. People, if you need a 3D structure, you definitely want the resolution, the higher the better. So why you are doing tomography, not single particle? Very likely is because you can't purify it, right? And then tomography samples, generally speaking, they're dirtier, they're more complex, noisier, and then they have other molecules in the background interfere your subtomogram averaging, right? Data processing. And very often, using my example, like the membrane fragment versus purified protein, generally speaking, the ice thickness is higher in membrane fractions comparing to purified membrane proteins, right? So the complex environment definitely going to play a role in limiting the resolution. And also ice thickness, complex environment, and also sample homogeneity. For most of the single particle complexes, you can biochemically purify it to reach the homogeneity so that you can average lots of particles, right? And then for most of the tomography projects, the samples are not as homogeneous as purified as single particles. But I see that as an advantage and a disadvantage. Advantage is you still have a little bit of their native environment in the sample. The disadvantage is really you have to try very hard to pick up, subset of it, right? For example, using the ribosome as inside the cell as one example. If you purify ribosome or in vitro reconstitute, for example, they are more homogeneous than what you can see inside the cell in terms of, summit composition, right? That's a very good point. Okay, can you think of anything else? So we talk about preferred orientation, ice thickness, homogeneity. What else? Anybody else? Okay. I can, one very obvious one, and then some of you actually mentioned it already, is the particle number. So you finish your your single particle modules already. So for an average single particle project, how many particles are we talking about?
Millions, I guess.
Yeah, right. For subtomogram averaging projects, using my glucan synthesis as one example, each tomogram, the best one, I probably have maybe 150 subtomograms I can use. And then some of the tomograms, I have nothing. I just have a piece of membrane with no donuts on it at all. So to reach the resolution of 14 to 15 aspirant resolution, I probably use like 3,000 particles. And then that's, a graduate students working very hard on it already. So the particle numbers is definitely something else we can think of. What else? Think about your single particle modules, what you learn from single particle modules. What else could be the limiting factor? One of you mentioned you have some single particle experience before. I don't remember who that person is. How about data processing? Single particle analysis. You basically have a very pure sample, very thin ice. You collect millions of particles and you average them to get your final map, probably 2.5 aspirant resolution. Think about a Cryo ET project like the one I did. So you actually have to correct many things of your raw tilt series to get your aligned map. And then from your aligned map, you back project aligned tilt series to generate 3D map. From 3D map, you can computationally cut out individual particles to align them to get a subtomogram average, right? Think about this. Anything you process, any step along the way, there are potentials for errors and imperfection. For example, I can't get a 100% perfect alignment, right? And then, so basically, I might have some errors inherited from data alignment to start with. And then from there, back projection, there are definitely some errors generated, passed along to the 3D map. And then eventually from there, you cut out individual particle aligning them. If your resolution limiting is limited to certain numbers from your subtomogram averages, no matter how many particles you can generate, how many particles you can include, you can't go beyond the resolution limit of your individual subtomogram averages, right? If you don't have that detail, high resolution information in your map, no matter how many particles you add in, you won't be able to reach better resolution. So nowadays, people started to improve subtomogram averaging resolution. People started to take kind of hybrid approach of single particle plus tomography. So they only collect small numbers of images per tilt series on a very limited tilting range. And then they actually potentially can treat each image in the tilt series as image by single particle approach. And then you process individual images instead of processing the whole 3D map. By doing that, you eliminate errors from alignment and you can more easily correct CTF. But I don't know, we can still call it tomography, subtomogram averaging or not. We probably need to come up with a new term for that. It's like in between of tomography versus single particle. Think about it to the very extreme. If you change the number of tilt series from, for example, 40 to 1, that's basically single particle. Right. Okay. So anybody else? Any other limitation factors? Those are very good topics you bring up. I'm gonna just bring here. Or anybody want to comment? I stopped sharing apparently, sorry. Okay. Yeah, so basically this is, several questions I think we can, think about when you're performing your own project, right? So for example, if you want to understand structure of your favorite protein complex, so you sometimes, depending on the nature of your sample, you can make the decision if, single particle or tomography followed by subtomogram averaging, which might be a better option for you. And then for data collection configurations, actually there are other parameters, other configurations we need to consider when you, performing your data analysis or data collection. That is, I didn't cover, I only cover maybe a very limited, very simple steps, not the, for example, imaging schema. And then those you probably want to talk to NCCAT staff to make a good decision. It's ultimately determined by your sample and also by your project goal. The missing match artifact, we can correct it, from imaging side, the single tilt versus dual axis tilt. That's one option to minimize it, to partially compensate for the missing match artifact by imaging. And then subtomogram averaging is probably mostly currently used in data processing to compensate for this artifact. And then for, factors limiting resolution of subtomogram averages, I think we cover many of them. And then I really like all of you, the comments. And then because I know you will, have a separate lectures on FIP, we have a separate lecture on subtomogram averaging. So I actually prepared some slides on, how you can identify objects from tomograms. Those are very information rich. So in very simple words, CLAM is the one people mostly using right now. But there are other ways, for example, computational approaches like template matching, or you can use biology, for example, performing mutagenesis to get rid of that protein to see what happens. There are other options. And then, for CLAM, if you want to apply CLAM to track down, your favorite target inside the cell, and then you probably want to consider, for example, the size of your target, how accurate you can really align a fluorescence image with a low MAC EM image for you to be able to confidently identify your target in the cell. And then those also requires, certain knowledge and certain consideration before but I'm just, in this lecture, I want you to know that option is out there. If you need it, I will be always happy to receive email from you to, have further discussion on that. I think that's all.